Thanks, Rachel, for that awesome introduction, and thank you all so much for being here. Whether on Zoom or in person, I really appreciate it. I'm going to take a deep dive under the sea today. Get away from that colder Nebraska, even though it's getting a bit warmer, which I'm happy about. But yeah, we'll go ahead and get started. Oops. Trying to get my clicker to work, so let's see. There we go. So I'll start with some background on nurse sharks. They're one of the most abundant shallow coastal water species that you can find in the Western Atlantic Ocean. And what this graph is showing is it's from a publication from 2023, and it was showing where most of the research on nurse sharks has been done. So you can see it's very focused in this red area, right over here throughout the Caribbean, Bahamas, and Florida. And this actually mentioned or included papers that only mentioned nurse sharks. So there haven't been a lot of major works done on nurse sharks in the Bahamas, but they have been mentioned in a lot of papers. They're in the order Electolobiformes, which are otherwise known as the carpet sharks. They share this order with species like whale sharks, wobegong sharks, zebra sharks, so very different morphology. And in the family Ginglymostoma, just a mouthful, I'll break it down. Ginglymo means hinge, and stoma means mouth. And that is referring to their the way they feed, which is they kind of do like this suck and crush kind of mechanism. And then also they were listed as vulnerable only recently in 2021. Before they were listed as data deficient, and that was because we didn't have enough information on them. There's a lot of information on them in the Florida Keys on their reproductive behavior, which I will dive into, and a lot of that was the inspiration behind some of this work that I'm doing as well. And then also, yeah, I said it right there, understudied outside the reproductive behavior. So I want to bring up a few key features of the nurse shark because they might look different than your typical shark you would think of. If you think of a shark, maybe you're thinking, I don't know, a bull shark, a white shark, something like that. These look very different. So the first thing is they have barbels, like the catfish in Nebraska here. It helps them find prey along the bottom because they are a benthic species that spend most of their time near the bottom. They have very thick skin, which helps them hide under rocky ledges, crevices to protect them from abrasion. They also have dorsal fins that are far back on their body, and that's so they can really hide under these coral heads, rock ledges, and really get under there. And also they have the absence of a lower lobe, which would make you think that they're not making these long distance migrations, but stay tuned. We'll see something quite interesting later. And then they can reach lengths of 3 meters long. So they are quite large. They can reach large sizes. The largest size was actually reported in Brazil at 316 centimeters. So yeah, quite large. And then this is an illustration of a juvenile right here, and I want to show the difference because I think the juveniles are also just absolutely adorable. They have little black spots all over their body that make them look like a little chocolate chip cookie. So the study area where most of this work comes from is where this red location marker is right there. And that's Bimini, the Bahamas. So if I go ahead and zoom in, there it is right there. It's composed of 2 different islands, South Bimini and North Bimini. And what makes this area unique is right here, all in this darker blue water, is the Gulf Stream. And typically, there's not a physical barrier there, but because of the fast current occurring here, the deep water, it can kind of act as a barrier to some species moving back and forth, but also it brings in a lot of nutrient-rich water to the area. And Bimini right here is right on the edge of where the Gulf Stream is, and the rest of this over here in this shallow blue water is the Great Bahama Bank, and that's only about 10 meters deep, all of that area. So it helps support in Bimini many different types of habitats like sand flats, seagrasses, mangroves, coral reefs, and allows a lot of different species to be able to occur in this area. Most of this work happened at the Bimini Shark Lab, where it was previously. They've been collecting data on many shark species since 1990. So they have these long-term data sets and accumulation of this data that is able to be maybe not analyzed right then, but maybe later a student would want to use it. And that's what I'm very thankful for, that I'm able to use a lot of the data that they've collected on nurse sharks. Because when they're going out and doing a lot of fishing methods, they would catch nurse sharks quite regularly, but no one was doing like a specific project on them. And so came into play. And first of all, we were looking at the data we had when I was still there, and there was a lot of recaptures in length and growth or length measurements for them. We thought, okay, what can we do with this? And this led to my publication that came out last year. And this was on the age and growth estimates for nurse sharks. And our goals for this were to find von Berlanti growth parameters for nurse sharks. What that means is it provides two parameters that shows, or three parameters really, but there's one called L infinity, and that's a theoretical maximum length that an individual can reach, and also gives you a K growth constant. And that basically is a constant that shows you how fast the shark is growing. So we wanted to get those because those are super important for including in fisheries assessment models. We obtained an empirical growth rate as well, and that's because we wanted to be able to compare a little bit to other studies. What complicates that is empirical growth rates are only based on the individuals you have in the population. It's an average of just all the growths. But those can be dependent on what size class you are catching. If you're catching ones that are more immature, they may be growing faster. And so if you're only looking at that part of the population, it can be hard to make comparisons, but we still obtain that to make comparisons. And then we wanted to obtain a maximum age estimate. So it was previously estimated that nurse sharks could only reach 25 years old, but it was theorized that they can actually reach quite larger ages because they've had them in aquariums and seen, okay, this nurse shark has been alive for 40 years. And also there's a study that had been done that came out a little bit before we published it, about 2 years, that through observation, they saw an individual that was probably a bit over 40 years old, but couldn't confirm that. It was estimated that they reach 15 years at maturity, that's about the range. And then females mature at 223 to 231 centimeters, males 214 centimeters about. And only empirical growth rates exist so far before the study in the Florida Keys in Brazil. And it showed fast growth, but they only included sexually immature individuals. Aging sharks can be quite difficult. So for a bony fish here, this is an example of an otolith, and you can actually count the growth rings in the otolith that they lay down actually similar to a tree annually. So if you take the otolith, which is the ear bone of a fish, for those that aren't familiar, you can take those out and actually count those rings, and then you can age the fish, and that's how old it is. But for sharks, they don't have those hard, bony structures that fish have because they're made of cartilage. And what people have done is they've actually taken the vertebral centra from sharks and counted the rings in there. But recent research from 2018, 2019 found that these rings may not be due to annual growth necessarily, but maybe more due to like physiological conditions, the environment they're in. So it may not be an accurate representation of that. So into some methods first, and I'll explain kind of what we did with our model, is that our sharks, they were captured mostly by hand capture and long line. And hand capture is exactly what it sounds like. Swim down to a ledge, just like these people are doing here. This is one of my co-authors on this paper. And you actually go find the nurse shark like under the ledge and you can grab it in its pectoral fins and just bring it to the surface. You can put it in a blue tub and then do all the workups with it. And that looks like this at the surface. So we could do this for small ones, but the very large ones would actually be worked up on the side of the boat. And I do have a picture of that later when I'm talking about one of the chapters of my thesis. And so the data that was taken for this were length measurements, which was a pre-caudal length, and that's the length measurement before the tail. And then the total length is the measurement including the tail as well. And then there was also a weight taken, a fin clip, and a muscle sample. And that's to look at different things like genetics, and also stable isotope samples and signatures, which tells you maybe like what the shark is eating, shows its trophic position. And then they were inserted with something called a pit tag, and this is exactly what your dog or cat have. And when you scan them, that's just like the same microchip that we put in sharks as well. And then they also get a dart tag. And what a dart tag looks like is kind of like this piece of spaghetti, basically. It goes on the outside of their body, and it has 6 digits that you can read and identify the shark. And what's really unique about this study is that we included some of the youngest and like smallest individuals in the population. This was the smallest nurse shark ever caught in Bimini, which was 28.6 centimeters, absolutely cute. And then also there were some individuals included that were like 280 centimeters, so quite long. So the methods we used captures that were greater than or equal to 90 days apart, and that was to be able to allow enough time to pass in order to see growth occur. And we use something called a Fabens method of the von Berlanti growth function. So normally, the von Berlanti growth function includes the component of age, but since it's harder to do that for sharks and we wanted to get like an age estimate, what we had is we had a length at first capture and a length at last capture. And so what we did is we rearranged this equation to actually allow us to put in that first capture and last capture, then estimate what the ages would be. And we did that all with this script that someone developed in 2022, which is this growth estimation script. So very thankful for their model that they developed, which is super helpful. And the big takeaway from this too is that we used Bayesian methods, so we were able to include prior information into our model, which was super important for this study because there were no von Berlanti growth parameters that existed yet for nurse sharks. So we didn't have those like go off of, so we had to use some prior information based on what's the largest nurse shark that's ever been captured, some stuff like that that we can input in there. And then this is just some information that we fed into the model as well. So what we found, these are all the captures of the nurse sharks around Bimini, whether they're captured by long line, which is this these circles here, hand capture, mostly down here is where we find the smaller ones, and then some other capture methods all around here. 91 individuals were used for analysis. There were a total of 137 that were recaptured, but we had to exclude them because of negative growth that we couldn't confirm whether the growth measurements were right or not, so we excluded those. They ranged in length from 48 to 252 centimeters that were used for this analysis, and the time at liberty ranged from like 1/4 of a year to almost 9 years. So this is the curve we obtained. The big takeaways that I want you to take from this is that the maximum theoretical length that we achieved for the nurse sharks was about 303 centimeters for Bimini. So that's what we estimated the size, the maximum size that they could reach there. This K growth constant we got was 0.04, which is very slow for fish. So we found out that they have some slow growth. And also the estimated maximum age we had for the individual in our population was 43 years, which extends the previous lifespan of nurse sharks. And based on those lengths and maturity that were estimated from a different study that like 223 centimeters for females and 214 for males, if we looked at that total length and like extrapolated that out to what their ages would be at maturity, it would be between like 20 to 30 years. Which is pretty interesting. I actually talked to some scientists about this and said, is this possible? Like, I don't know, that seems like pretty cool. And they said, actually, we haven't been able to confirm a nurse shark that is mature before 20. And so, yeah, very possible. So some implications from this. It was the first study to obtain these von Berlanti growth parameters for nurse sharks and indicates that nurse sharks are slow growing and long lived, something that was not previously thought for them. And then it extends the previous lifespan of nurse sharks as well. So yeah, now that we have these parameters, other researchers in different areas and regions can actually use these to then compare between populations, because these estimates, instead of just like an average growth rate of all the individuals in the population, are more comparable because they actually correct for some of the gear selectivity issues. And so yeah, people can compare them with other studies now. So that brings me to my master's thesis now and what I'll be working on here. There's that cute little nurse shark. So some background on the reproduction, because a lot of my master's has to do with the reproduction, genetics, and what exactly is going on in Bimini in the Bahamas with that. So some background is that the reproductive behavior is like the most studied of any elasmobranch, which is sharks, rays, skates, all of those. And so yeah, and that's because of these researchers that have been in an area of the Florida Keys, the dry tortugas, and they've been doing research there for like 35 years or over 35 years, and they're able to observe them actually mating and see their behavior there because they do it in such shallow water. This is very shallow water up here, and that's where they'll do it, and they can actually observe it. And what they've seen from these observations is that these nurse sharks that mate in this area have mating site fidelity, where the females return every 2 to 3 years, mostly every 2 years, but the males return every year to the specific site. And they've seen that from tagging information, where they can either scan them or they have a visual tag and they say, oh, well, there's that individual again. Okay, they're back. And they've observed some individuals coming back like for the whole entire time that they've been studying this area, so like 35 years. And a puppy in the nursery grounds is yet to be identified. In this area, they've looked and seen if they can see like any neonates, which are newborn nurse sharks, and they've only seen some juveniles. So they see some juveniles around, but they haven't found any like newborn nurse sharks spending their time there, or juveniles really seeming to use this area. And so they haven't been able to establish it as a puppy or nursery grounds per se. And as I was mentioning earlier, because nurse sharks mate in such shallow water, you can actually observe them right there, which you can learn a lot from. And because they were taking a lot of genetic studies in the Florida Keys of them too, all the research there has found out that nurse sharks have polyandrous and polygynous mating systems. And so the males will mate multiple times and the females will mate multiple times. And there's multiple paternity actually in a single litter. In one litter, for nurse sharks, I think they found up to 7 fathers, I think it was, in one litter, which is pretty interesting. And they found that females have this avoidance behavior too. If they don't want to mate, they'll actually like beach themselves almost to like get away from the males. Or like actually sperm storage as well, females do, which maybe that's how there's multiple paternity as well. Some of the sperm that they've stored over time, maybe they'll like, no one really understands the mechanism, but maybe that's how there's some multiple paternity. Maybe they don't necessarily that mating period mate with 7 males, but they have some sperm stored up. And also there's some male dominance hierarchy too that they've observed. They've seen that like some males will actually seem to go like block other males from mating and allow like one male to mate. And they don't really understand that yet. They think it could be like maybe they're related somehow, or maybe there is just a dominant male and he's the one that gets to mate the most. So in my study for chapter 1, we're starting more on the individual fine scale level. And what we'll be looking at is the local phylogenetic population structure in Bimini, because we have almost 300 samples of nurse sharks just from Bimini from 1996 to 2019, where we will observe, okay, in Bimini, what's going on then? Are we seeing like polyandry, polygynous mating systems as well, multiple paternity? What are we seeing? And what we'll be doing for that is actually, we'll be using some genomic data, and we'll be looking at SNPs, which is a single nucleotide polymorphism. What that is if you're looking at the genomic data of one individual and then of a second individual, it'll be this one change in like these base pairs right here. So that change. Some of the goals of this is to determine if Bimini is a puppy or nursery grounds. It's been theorized for a very long time that Bimini is an area where that happens because that 28.6 centimeter was a neonate, one that was caught. That's actually like around the size they are at birth. And they continually find under these ledges a lot of like small juveniles spending their time under there, but we haven't been able to confirm yet if like, are those siblings hiding under there? How long they spend it? So some of that genetic data will help us see some of that information. And then we want to see if adult nurse sharks display phylopatry as well. And we'll be able to see this based on how many times like a male or female is showing up in the genetic data of like the offspring, and see like, okay, are we seeing that they seem to have a litter every 2 years? How's that working? And then we'll identify parent offspring or sibling pairs as well. I mentioned a bit earlier. And then we'll investigate polyandry and parentage with that as well. And so I've started part of this, or oh my goodness, hold on, close your eyes. So I started some of this a year ago, and it's actually in collaboration a little bit with the Chicago Field Museum as well. And that's where I started it a year ago. And I started with the Bimini samples. I began extracting DNA. We have 296 tissues from Bimini specifically. And so that's what I'm using for this first part of my analysis. And then we're going to amplify these actually afterwards. So I'm going to Chicago in like 2 weeks. I'll bring all these samples back here, and then we're going to be completing the rest of the part here. And that's what we'll be doing PCR to amplify this genomic data to get a lot of reads. And then we'll end up sequencing it using a double digest restriction site associated DNA sequencing, DDRAD. And what DDRAD actually is good for is it can reveal like fine scale population structure and elasmobranchs, and which is exactly what we kind of want to look at. And then once we get that information back, we'll filter and check the quality of our sequence data. And then we'll analyze using a program called Colony, and Colony can help assign like sibship and parentage, and it's quite good at that. So that brings me to chapter 2. This chapter has actually come in a bit later than the other ones, and that's because we actually found some really interesting information from the nurse shark acoustic data that we had. So there's some acoustic data on nurse sharks, which is like tagging, acoustic telemetry. They have like some internal tags in them, and they last for 10 years. And so it's going on like the 10th year that the program started. So this started as a PhD students project, and they were looking at many different shark species in Bimini and where their habitat use was, basically all around the area. So I'm thankful nurse sharks were included, because now this gives us some really interesting information. Because we want to see what are the movement patterns associated with like reproduction, site fidelity, and phylopatry migration. And the goals from this are, I just want to determine the movements. Where are they going? Are they leaving Bimini? Are they staying close to Bimini? Are they going in other areas of the Bahamas? Because I think this will help provide a good story with the genetic data as well, to kind of show like, okay, we know from this genetic signature that this female has phylopatry here, but what can we see too? Because all the individuals that were acoustically tagged, we have genetic data for as well. We want to examine the degree of residency/migration to and from Bimini, see if they're spending most of their time in Bimini or leaving, making migrations, and assess any effects of sex and seasonality on these movement patterns to try and see, okay, what's driving some of the movement or not movement. So with the tagging, in this picture is pretty cool. This was the first nurse shark that they ever acoustically tagged in 2015. It was around March. There was 20 adults tagged, 11 males and 9 females from 2015 to 2017, and they were tagged with these acoustic tags right here. This is what it looks like. And how it works is there's an array of these receivers under the water all around. I'll show you the Bimini array specifically next. And these tags that are internal inside the shark can communicate with these receivers when it swims past it. And they have varying ranges. There's 65 receiver sites around Bimini, so this is what the array looks like here. And they have a 500 to 958 meter detection range, and that depends on the habitat that it's in. So if there's a receiver in a sand flat where there's no obstruction around, it can actually detect a shark that swims within the vicinity pretty far. But say it's in an area where there's mangroves, there's a lot of entanglement around, the detection range can be a little bit less. And this array was designed to have no overlap. But that was before they really did rigorous detection range testing. They found, okay, there's a little bit of overlap in some areas, which is okay, because then you can actually do triangulation and figure out pinpoint exactly where a shark was. And then they occupy all different types of habitat. And this is what a receiver looks like under the water. So this will be like a tagged great hammerhead here, swimming within this. And what the tag will do, it'll communicate with this receiver, download all the information. And some of these tags actually have like temperature, pressure sensors as well. So you can actually see like maybe what depth and all of this, what the shark swimming at. So an advantage of this is that these receivers are actually part of a larger network. And so the ones that are used in Bimini are the same ones used all along like the east coast of Florida, like all the way up to like South Carolina, like I don't even know where this is, like North Carolina, and then Gulf of Mexico, other areas of the Bahamas. And so what that means is that if our shark leaves Bimini, it can be detected in like Florida or somewhere else as well. And there's a big collaboration with this, which is really cool, because if we weren't doing all these collaborations, we wouldn't be able to figure out the movements of the nurse sharks and only what they're doing in Bimini. There was a study done in 2022, and this was the PhD student that was working on this project, and he determined the general and core use areas of the nurse sharks specifically around Bimini. And these darker blue areas are these more core use areas. As you can see, they're quite widespread all around Bimini. There are some receivers over here, but a bit more limited. So they are definitely still in this area. I've seen them over here, but maybe not being detected as much. And this was the result from the acoustic data when I extrapolated it out from Bimini, which is really, really interesting to me, because it's been reported that these sharks have very specific site fidelity, restricted movements, but this does not really show that. You can see that one nurse shark like traveled all the way up here, and 20 sharks, so all the sharks tagged, were actually detected in Bimini throughout the study. This is where most of the detections were. There was an area in Jupiter, Florida, where 8 sharks were detected, and there was quite a bit of detections over there. And what's really, really fascinating to me is that there was one female, and if you only measure the straight line distance, I need to actually like map out her whole journey. But with the straight line distance, it's 773.5 kilometers, which is the largest migration recorded for a nurse shark to date, which is pretty cool. I don't want to get too bogged down with the details here, but I'll point out a few key things. So this is the residency index, and basically what this is calculating is the number of days total that the shark was detected over the whole array, and then it took the amount of time it was detected in specific areas to kind of get that resonancy index. So what I was looking at was the one for Bimini. How many days were they detected in Bimini relative to the amount of times they were detected throughout the whole time? And 11 individuals were actually only detected in Bimini. They were never detected anywhere else, and so about a little more than half of them. What was cool was 2 individuals actually spent less than half their time in Bimini. They prefer to spend their time in other places. I want to break that down eventually and see what time of the year maybe were they tagged in Bimini. Would they just be passing through doing something? Because what I want to eventually look at is, leads me into this, is focus here on the Bimini array. That's the dominant color in this plot right here. And this is for each individual. So these are the 20 individuals over here. This is the time of year. And so as you can see, there's a dominant color of Bimini here, and you can see the individuals that spent most of their time. And it actually shows support for something called partial migration, and this is on the population level, which means that only some individuals of the population are migrating while others are choosing not to. That's basically just what that means, and you can see that here because some individuals never migrated from Bimini, but some did, and like this one's really interesting, had this repeated migration to this area. And I noticed when I was looking at this, where are these breaks right here? And so I drew straight lines and they seem to match up a lot of them. And when I actually looked at like the specific dates, it's around when they actually mate in the Florida Keys. And so I was like, huh, okay, that's interesting. I want to look more into that and see, okay, like, are they, like, is there maybe like a specific area, but they could just be going outside of the array. But it's interesting that it all lines up. Maybe there's a specific like mating area for nurse sharks in Bimini. Not quite sure, but yeah, I want to see if I can look any more into that. That's correct. So summary of this data so far then is that it shows that nurse sharks are definitely capable of making long distance migrations, and they can pass the Gulf Stream, which has typically been thought of as a very major barrier to them. And then most individuals are residents in Bimini, so most are choosing to spend their time in Bimini all the time. Shows support for partial migration, some individuals leaving the population, coming back, and some just staying in Bimini. And then it shows that site fidelity / final patry is also plausible because of that one individual that was spending time in Bimini, going somewhere else, coming back to Bimini, going somewhere else. Kind of shows maybe they have an attachment to Bimini specifically though. So some future directions for this chapter is I want to look at where individuals, and so there is that paper where it's looking at the whole habitat core use of these nurse sharks, but I want to look more individually and also combine it a bit with the genetic data to be like, okay, are we finding in this area that a lot of these pups are related to this individual that was detected here? Kind of some of that information. And then I want to examine the effects of like sex and season on the individual movements and migrations and see if that's impacting their timing of migration or anything like that. That brings me to chapter 3, and this is a big reason why we wanted to include the acoustic data as well, because we think it helps create like a really good story for where these nurse sharks are going, what they're doing, and what we want to look at is more population level, so moving from the individual to population and assess the population connectivity as a whole in the different islands of the Bahamas and where we could get samples from. So the goals of this are to determine if nurse sharks in the Bahamas comprise like a single population, distinct populations, see if in different islands, like they're very genetically different from the ones in Bimini, or if they seem to all be reproducing together or what's going on there. Examine genetic differentiation and like the gene flow among the islands as well, using some parameters. Evaluate potential barriers for gene flow as well, as there may be one population that seems to be super separated than the other ones, and maybe what could a barrier to that be? Try and see that. And then also eventually combine this with acoustic data to create a network analysis of where these nurse sharks are going, where they're spending most of their time. I'll talk a bit more about that. So I want to give some background on this. There's been a few papers looking at the genetics of nurse sharks, but one looking at their population connectivity. They use samples from Brazil, Bimini, Florida, and an area of the Eastern Pacific. So I can't remember off of what island it was, but it was in the Caribbean, and then Belize. They used samples as well. And what they found is among all of these populations, they found that they were very genetically like depauperate. So they had very low genetic differentiation among the different populations, except the one in the Eastern Pacific, which has now been defined as a separate species. But before this paper, when this paper came out, before they defined that. And there's this weird like discordance with, they have very large population numbers, but they have low genetic differentiation. So what they think that's because of is indicative of a recent expansion of their populations. And then it also, in this paper, they assume nurse sharks do not disperse through open water, which we've seen now that they are able to do. And males and females, they found, do not have differential phylopatry. And that's something that we're still going to investigate and see, okay, does it seem to be different for like the males and females in Bimini? And they also use microsatellite loci and mitochondrial DNA, and we'll be using something different, and I'll point that out. So our methods, we have about 360 samples total, including those Bimini samples. So we're including other islands in the Bahamas, which are the Baylie Islands, West Andros, and the Upper Exuma. And I'll point these out real quick. So there's Bimini right there. There's the Baylie Islands, and then West Andros, and then the Upper Exumas. So all from these areas. And actually, we have a few samples from the Rocas Atoll in Brazil as well, because the Bimini Shark Lab actually used to go to Brazil to do some sampling there, and they actually collected a few nurse shark samples. So we're going to use those as well and see if they show a lot of genetic differentiation from the populations there. And the methods we'll be using is that single nucleotide polymorphism again, where there's that single base change. They get to actually determine the connectivity and population differentiation. And the reason we're using these is because they can actually reveal like more cryptic populations that are sometimes not detected by like the mitochondrial DNA. And it could be important for nurse sharks that has previously been reported not having that much differentiation. So that's why we'll be using that method. And then we'll analyze them using programs such as structure, splits tree, and using the principal components analysis. And what that helps show is like it actually puts like the genetic data into different clusters kind of and shows like where they are and shows if they're pretty different. If they're all clustered together, that means usually there's not as much genetic differentiation. But if we see them separated out by island, that could indicate some differentiation there. And we'll compute some fixation indexes, so FST values, to determine the level of genetic differentiation too between these different islands. And then, yeah, eventually generating this network analysis that'll combine this movement genetic data using R packages such as iGraph and Circlize. And so in summary, for the master's thesis, the first chapter will be more individual, look at this fine scale populations, what's going on there, examine this low profile phylopatry population structure, sibship, parentship, what's going on specifically in Bimini. Second chapter, we'll analyze these movement patterns and see if they have any correlation with some of the genetic data we're seeing, and just define some of their movements too, since that has the longest migration to date. And then we'll finish by looking more at this population level of the population connectivity between the different islands in the Bahamas and that one area of Brazil. So I want to point out some future directions as well. This won't be part of masters, but I just wanted to show it too. This is a project actually that started in 2022, towards the end of it, and we've been collecting blood samples from nurse sharks, all different size classes, and taking ultrasounds of some of the older individuals. And what's really cool is that from these blood samples, you can look at like reproductive hormones and see certain stages. Okay, what reproductive hormones are like more present, maybe which ones are driving like sexual maturity, reproduction. And what's cool about it is that no studies to date have combined this with ultrasound data. So what will be really cool is we will be able to confirm some pregnant females and then compare that with like the reproductive hormones that we're finding and see, okay, this is what's important when they're pregnant. What does it look like when they're not pregnant? Which ones are playing more of a role? And then I'll finish by showing you an ultrasound that I took in 2022 of a pregnant nurse shark. You kind of have to use your abstract mind a little bit, but what it looks like is if you like go to the store and you buy like a tuna steak and it has like the vertebrae there like in the center and kind of like all the muscle on the outside, that's what it looks like for this because it's taking a cross section of the nurse shark. So you can't get like their face or anything. So I'll show you that right now. And so you'll see them kind of up here in a bit. There they are. There's two right there, three. And that one you can kind of see it moving a bit. And there's another really good shot coming. And then a few moving there, so that'll be cool and in the future. I want to give some acknowledgements to a lot of the organizations, institutions that are helping support this research. And a special thanks to the 1,000+ staff and volunteers that have collected field work during the duration of this project, and for having those long-term data sets that we'll be able to analyze now. Thank you to my advisory and committee members, Dr. Renee Martin, Dr. Sarah Sonstagen, and Dr. Rebecca Varney. Hi, Rebecca. I think I saw her on here. And then all the funding and all of the media that they allow me to use for this presentation as well, and the illustrations. And with that, are there any questions? And for you guys on Zoom, I will repeat the question so you all can hear it. And you can also submit questions from Zoom as well. I'll look at the chat. Yes.
Thank you so much for a great presentation.
Yeah.
I feel like if the PhD, a PhD work is of a master's work.
Yeah. Yeah, I am so a good advantage of this is that all the field work has been collected. So that's definitely helped a lot. And I started it a year ago. And so I've been able to do a lot of the background and yeah, have all that field work collected. So I we'll see the time frame I can I mean, well, it tells you it's like I'm confident that I can finish it on time, but we'll see as well.
Yeah. So to that, I wanted to know how many species of nurse shark are circulating in the area.
That's a great question. And so the question was, how many nurse sharks or species are circulating in the area of Bimini? So just this one. Yep, just this one, which is Ginglymostoma cirratum, and it's the Atlantic nurse shark. And actually, it's the only erectoloba form that can reliably be found in Bimini as well. Sometimes there's a whale shark that passes through, but very rarely. So, but yeah, so there's actually about 5 species of nurse sharks, but the rest of them are found in like the Pacific, Indo-Pacific. Yeah, nurse sharks are the only ones found in the Western Atlantic. So. Yeah.
So regarding migration of these nurse sharks,
yes,
maybe you already mentioned this, but do you think there are any like environmental drivers of migration, and do you think you'll be able to incorporate any of those drivers in your models?
Yeah, so the question was, pertaining to the migrations, do we think there are any environmental drivers that are driving this migration? So yes, I think so. We have some information on like seasonality, and also like we can pull like some temperature data as well and see some of that. And so I'm going to incorporate that into some models and see if it has anything to do with like if the weather gets warmer, is that timing them to migrate somewhere else? And so we will look at that. Yes, and I do think the ones that are migrating, they're being driven by some environmental cue, because that's pretty standard in a lot of elasmobranchs. Usually there's some kind of cue that'll have them migrate. So yes.
When you were talking about the site fidelity, you said the females only come back every about 3 years or so, but the males are yearly.
Yeah.
Does that mean, are the females not mating every year, or might they travel to a different site? And
That's a great question. So the question was, for when I was talking about the nurse sharks returning to that area in the dry tortugas, and the females only returning every 2 to 3 years, is that because they are going somewhere else to mate during that, or they're taking time off? And the answer for that is they are taking time off. And so they reproduce every 2 to 3 years is what they've actually seen from some genetic data from the Florida Keys. And actually, I want to look at some of that too in Bimini and see like, okay, because they've actually done extensive studies for this for lemon sharks. I think Kevin's on there. I think I saw Kevin. And he's done most of the work for the lemon sharks. And he's seen that lemon sharks have biennial reproduction because he's seen like there's been, in the offspring, there's been a signature of like the females every 2 years in them. And so with the nurse shark data, I'm hoping to see like some of that as well. But yeah, short answer is that they're reproducing every 2 years and returning every 2 years. And so that's kind of timing up. And in the time in between that, there was a paper looking at their movements, and they actually like don't even go to that mating site when they're not breeding that year. So yeah. Cool, good question. Any other questions? Any questions on the chat too? I can see the chat.
I have a follow-up from you.
Yeah.
Well, you had so much great information in this presentation. So you kind of mentioned offhandedly that a female, she doesn't like the way a certain male's looking, she'll beach herself before mating. Is there some kind of female mate preference that she has?
Yeah, that's a great question. So the question was, is there some sort of female like choice involved in reproduction? And I think so. Like they theorize that possibly there is, and with sharks it's quite hard to see, and they think it has to do more with like their sperm storage and when they actually decide to use like certain sperm. But anyway, they do have avoidance tactics too. And that's where like when they're beaching themselves or don't want to mate, and that's because mating can be quite energetically costly for sharks in general, because like males like bite onto the pectoral fins or like backs of females and they can get like super cut up. And so it could be that they just mated too and they're like, no, I'm tired, like beaching themselves. But yeah, so I it's hard to say exactly what that female choice is, but there's some level. Yeah, yeah, of course. Any other questions? Any questions on the chat?
Any questions?
Oh, yeah.
So earlier you said that it was found that lifespan is actually higher than what was thought.
Yeah.
Do you think it's possible that lifespan is changing?
Yes, and I know that they, oh, that, do you mean like changing over time?
Yeah.
Oh, interesting, okay, so the question was, do I think that the lifespan of nurse sharks has changed over time and they possibly used to be shorter and are longer now? I can't say for certain, but I don't think so. I think they possibly lived that long and they just didn't know, because they used to keep nurse sharks like in labs a lot, and they'd work with them in labs and like aquariums, and maybe then like they weren't like living as long because we didn't know how to take care of them in aquariums as much. It's hard to actually confirm that in the wild because there weren't these studies set in place that have been done for like 3 decades. Once these researchers were there looking at them for like 3 decades, aquariums got better. They saw they're actually able to live a bit longer, and I've actually spoken to someone, where was it? An aquarium in Maryland, I think, and they have a nurse shark that's lived to be 50. I think it's 50 right now. So yeah, they can get to be old. Cool. All right, well, I think that's it. Yeah, thank you so much, everyone. Everyone get some pizza if you haven't gotten some yet. Thanks to everyone on Zoom. Thank you so much for coming, and I appreciate it.
Next week at 11, so it's at 11.
Thank you.